Talking about the cover crop, and I probably approach these things a little bit different than other people. Appreciate the research and data that Paul has to present, and we'll see how I come out with what I want to talk about. But soil carbon is linked to all measures of soil health. And the carbon, a lot of people talk about carbon. It's so important for carbon. And what is carbon in the soil, to begin with, is kind of the same carbon that those donuts, that wherever they are, we eat a lot of carbon. We call it food. That's what the carbon is in the soil, is food for the microbes that Paul talked about and Dan, everybody talked about. And there's three things that affects. That's the physical is the first one I look at, is because we talked about the water and water intake, is getting the soil structure so that it takes water better. The chemical is what I'm always interested in, the fertility, because we have to have nutrients there to get the biological activity to go, as Paul talked about. And then the biological. So we're going to talk about all those kind of things. And so what is soil carbon? Number one is it's a term that we use, exudates. It's plant roots leak out compounds. And Rick Haney from Temple, Texas, he told us that there are about 95 different compounds that roots exude out or leak out of their root system that feed microbes. And different plants leak out different compounds. So to increase that microbial population, we need to have a diverse of plants. And then, of course, enzymes, that's what helps break down the carbon, all the food or the residue, the plant roots, whatever you want to call all those things that the microbes are eating. Enzymes help to break those down. And then we've got the soil microbes, of course, and then decomposing plant roots. We got the plant roots in the soil. Now that's food for the microbes. But the most important food is the stuff that's leaked out of those roots as the plants are growing. So it's important to think about how Mother Nature handled our land before we got here. And that was, she had something growing from the time it started warming up in the spring until it froze solid in the fall. We have a crop growing from April 15th or April 20th to October 1st or whatever it is. And then we have maybe soybeans growing from May 1st to September 30th. And so that we got a lot of time when there aren't things growing. That's what the cover crop then is to think about supplying food for the microbes and decomposing plant roots. Decomposing plant residue, we talk about that. The soluble organic compounds that are food for the microbes, and then the soil organic matter. It all builds the soil organic matter, as Dan showed in his slide. Cover crops, the carbon, cover crops, soil health. Cover crops feed the microbes. And how many microbes are there? Well, I got a slide to show that a little later. These things, you can't see them. It's hard to understand, but we need to think about that. Cocktail cover crops diversify the microbial population. A mix of cover crops, and Paul showed that a little bit, but I really think the best cover crop is kind of mixing. The big boys say over 10, but Paul had like 2 or 3 or 4 different things in there, but try to mix that up. And then more diverse cropping is needed for soil health. And I know this is corn, soybean area. Saline County, where my home is, I grew up in Saline County. It's pretty much a corn, soybean, but we do have more wheat in the rotation there than maybe you do up here. But what other crops can you grow in there? And I talked to my tenant, and I said, I'd like to put milo in the rotation. And he said, oh, that itches. I kind of ended the conversation, but so I'm driving home, and I'm thinking now, he's got that great big combine, it's air-conditioned cab. What in the hell is he worried about? But it thinks about when my dad was combining, and Dad always combined milo when I was in high school, so I didn't get involved in that part of it. But I think, let's think about some diversity that maybe we can get in this thing. And then reduce nitrogen fertilizer, because microbes and legumes fix nitrogen from the air. We had a discussion last week up at Pierre, South Dakota with Dwayne Beck and his board and some people in talking about How does plants get the nitrogen out of the air? No, it's the nitrogen in the air, in the soil. It's where the plants get it, where the microbes get it, and the rhizobia. And that soil structure, that physical part we talk about, if we have good, stable, water-stable aggregates, and the water can flow in here, and we keep those aggregates, now we have a passage for air. As the plants use the water, air comes in, and as we get rain, water goes in and pushes air out. You know, that kind of a thing. And I finally figured out that all our soil is, when you think about it, is a great big lung. Breathing in air as the plants use water, and breathing out carbon dioxide as the plants, as we get water going back in, irrigation or rainfall. So it's just one of those things, and this is kind of my soil. I dug up, took a picture of it, because all that white mycelium hyphae there that we have, and for some people I can say that's there's my mycorrhiza, but I know it's another kind of fungus. It's not really mycorrhiza, but it makes it look the same. And that white stuff you can see visibly there, the hyphae for the mycorrhiza is microscopic. And Christine Nichols told us one time down in Marysville that in 1 gram of soil, which is about 3/8 of an inch cubed, if you can imagine how small that is, 3/8 of an inch cubed, 1 gram has 100 meters of mycorrhiza hyphae. 100 meters is the length of a football field. You know, and of course, how in the heck do you know that? You kind of thing. But you know, with microscopes you can see a lot of stuff. And if we are doing a lot of tillage and maybe starving the a part of the times, mycorrhiza doesn't like it, and they'll go away pretty fast. And that's what Paul was showing with that flooded field. It had flooded. The mycorrhiza is the thing that dies. And now the mycorrhiza fungi is what's needed to bring especially phosphorus, but all the nutrients to the plant. And it's on that interchange of the plant roots and with the different plants growing out there. You know, they say, well, the roots, the plants can trade water and nutrients and energy and those kind of things. And I had a hard time understanding how those roots could connect each other and how the plants would do that. And finally, I've read in a book that some guy had carbon-14, which is radioactive carbon, put around this fir tree, and is trying to figure out how the carbon is transferred within that fir tree. And in his research, he found an aspen tree down slope that had carbon-14 in it. And then it made me realize it's the mycorrhiza that connects those tree roots that is trading with the plants, the carbon and whatever else might be, nutrients and water. And so you think about in the mycorrhiza in the soil, how we can get really efficient in some nutrient uptake is by having that mycorrhiza. And that's what it increases with cover crops, but you have to have kind of no-till farming idea before you can really get the mycorrhiza to grow. But it's just a way to show, and then the granular structure that I show there. Now, Paul talked about water, and so this is one of our soil types on our farm, and if you're a good soil scientist, you know Muracet loam is one of the best soils we got in the state. And but the water holding capacity of that Muracet loam is 9 to 10 or almost 11 inches. So it says 10 inches of water. That's all that soil holds. You got a 50-gallon diesel fuel tank on the tractor. How much diesel fuel does it hold? 50 gallons. How much do you use out of it, and how much do you put back in? And we have a tendency to think that this soil holds more than 10 inches of water, but it doesn't. And so when I look at my cover crop, and I'm planting it in right now into wheat stubble mainly, but this amount of rainfall that was received in Saline County from July to next April when we plant corn. I got 21 inches of water on the average that is received, and if I have wheat stubble in bare soil, I'm wasting 10 inches of water. If my soil was dry when I started the thing. So why not have a cover crop growing to utilize that water? Now you could take July, August, and September off of there, So that'd be 8, say about 10 inches, you still got 10 inches of water for a cover crop after corn. And then I looked in Saunders County, and Saunders County gets 4 inches more rainfall than Saline County on the average. And this is looking at Nebraska rain. You can find all these things out in there. So it's a matter of, you worry about the cover crop using water, and I'm worrying about, why do I not use my water? Think about it, kind of make some plans on how you can utilize that water better. This is a cover crop on our farm this year, planted in the wheat stubble. And I don't have the time on there, but you can see there's some broadleafs down in there. And of course, the sorghums are growing on top. And there's I had 11 or 12 different species in there. And then in the fall later, it grew up to be pretty big. And I have a grandson who's out there before it froze, and he was standing by one of those sedan stalks. He's 6'5" and is about 4 feet taller than him. And he was so disappointed on the first frost that tall stuff fell over. He went down there to do some hunting, and he said, well, it fell over. Well, yeah, that's good that it fell over. And the other story was he got a 4x5 buck that him and his buddy were walking back through the cover crop from their tree stand, and a buck come out into the cover crop and stood there sideways to him and show off, and he got him. But so he thinks cover crops is pretty good. He's a student at UNL. So the cover crop has nutrients in it, and how many of those nutrients are available for the next crop is what we're trying to decide. And I just have an example here of analysis, and the one that we started throwing into these is carbon. The one nutrient we never talk about is carbon, and 42% of the plant in this case is carbon. And so think about that a little bit. 1.6% nitrogen, phosphorus 0.22%, potassium 1.33%, and sulfur 0.31. So it's pretty good to cover crop nutrients here. So just for 1 ton of that hay, dry matter, there's 840 pounds of carbon in there, and then 32 pounds of nitrogen, 10 pounds of phosphorus, 32 pounds of potassium. Now, sulfur is 6.2 and a little bit of zinc. But these are things that are being recycled up to the top. And Dan showed that top 2 inches is 0 to 2 on his organic matter. There's also, when you have the organic matter there, you have all those nutrients in there also. And there's more here besides what I put in the slide. So just to give you an idea how you might be able to calculate that. Well, here's how you do that. If you're really curious about your cover crop and nutrient cycling, is it gets you and probably, Jim, it'd be better to use a mower like this, a little hedge clipper with a gasoline engine on it, than try and do that by hand. But clip an area, a yard square, inside of there, and pick all that stuff up. Try stuff it into a bag, and you can send it to Ward Laboratories. We'll weigh that. We'll chop it up. We'll take a sample out, dry it, and analyze it for the nutrients, and calculate pounds of nutrients per acre based on the yield of the production that you had. And if you're, just an idea of what, if you had like knee-high cover crop, would you have a ton of hay? A little bit, but how do you decide how much that is? Well, we probably had about 4 tons where that stuff was so tall and in that mixture. But with rye growing in the spring, maybe you have a half a ton. But if you had 30 pounds of nitrogen in that, you're going to use it, got your nitrogen fertilizer rate 30 pounds. That's why you want to have cover crop to recycle nutrients. Now, are you going to use those nutrients? Or what, that's the other part of this cover crop thing you got to think about. And here's an example of a cover crop. This is in the wheat stubble. And October 11th and November 5th, of course, things are growing all that time. And the carbon, 1,800 to 2,500 pounds of carbon in there. Nitrogen, 119, 161. If I could recycle 161 pounds of nitrogen in that cover crop, how much can I reduce my nitrogen on my corn next year? Those are the kind of questions, and that's where the value of value of the cover crop seed costs and that kind of thing. But if I can use 80 pounds of that nitrogen, man, that'd be a great savings. At, 35 cents a pound, it's what, close to $30 an acre, just in nitrogen savings. Then the phosphorus, you just recycle these back up in potassium and sulfur. But the nitrogen is the one that I think is the most important. How much can I cut my nitrogen by having that cover crop? And it's a big question that we're still learning how to do that. And I have lots of ideas, but I don't have very many answers to some of these things. But it's a kind of thing. It's a school. You should be thinking about what you might be able to capture. And I'll just say it this way: it would be nice to be able to do some of these things without somebody saying that you got to buy something to help you. Maybe you could help yourself by doing some of these things. So this is just photosynthesis. I think you all know this. Sunlight, carbon dioxide, and water captures the energy. In the case of food, we call that energy calories. In the case of maybe wood, you call it board feet. In alcohol, and if you're driving a car, whatever that miles per gallon, and that's all energy, and that's photosynthesis. That's what the plants do. That's what you guys are doing. You're capturing sunlight and then selling it as energy. But when we get this energy, and then when we eat that donut, we do respiration. We take this sugar in, or carbohydrate. I better call it carbohydrate instead of sugar. We take this carbohydrate in, and then you breathe in oxygen, and you breathe out carbon dioxide, and what else? When it's cold outside, you can see your breath. That's water. So think about this now. In this respiration is residue decomposition. So when the microbes are breaking the residue down, they're eating this carbohydrate, and they're breathing out carbon dioxide, and they're releasing water. So if I had my cover crop or my residue decomposing in the summertime, I would be getting carbon dioxide to feed my plant, and I'd be getting water to feed my plant out of that respiration. It's just one of those things that we don't talk about very much. And Neil Dennis, that's a mob grazer up in Manitoba, he says his grass greens up before anybody else's in the neighborhood in Manitoba, and because he bale feeds out on his grasses. And I said, I thought a little bit, is that respiration? He said, yes. And so think about that a little bit. Can we warm our soil up a little faster with a cover crop that starts to decompose and is doing respiration? Because the other part of respiration, why do we eat food? For the energy. And that's to keep our bodies warm. Maintain our temperature. So the other part of the respiration, that energy that's captured is released as heat. So with a good active soil, your soils are warmer. I don't know how much, but these are things I want you to think about. So that's just one of those exciting things. So that carbon, here's the 200 bushel corn, 4,000 pounds of carbon in the grain. So when you take that grain off the field, you're hauling off 2 tons of carbon with that 200 bushels of corn. And then I say how much carbon dioxide, because it's fed as carbon dioxide. And I have to make a comment that carbon dioxide is not a pollutant, it's a plant food. The more carbon dioxide we have, the higher our yields are going to be. So in the corn stalks, this is the part we leave in the field. We have 5.6 tons of 200 bushel corn. There's 2 tons of carbon, or 4,000 pounds. And how do I get rid of that residue? Everybody's worrying about how to get rid of that residue. And when you turned off a 77 coming over here, I didn't see a disc field from when I was driving this morning. So we're getting the idea. Because Jerry Hatfield at Ames, Iowa, with the research over there, he said in Iowa, after corn harvest, the carbon dioxide goes up 40 parts per million in the atmosphere in Iowa. Because guys are doing tillage and breaking all that stock residue down to the carbon dioxide when there's nothing growing. So here's what the idea is. To get rid of that residue while the next crop is growing, so I can capture that carbon dioxide. So you know, these are the things that are doing all that work for us. So we talked about some of the physical, a little bit of the chemical, and now the biological. And these things are real small. Up here, 1 million to 10 million per gram. Remember that 3/8 of an inch cube? 10 million microbes in there, bacteria and fungi. It's just amazing how this stuff does that stuff. And they have different shapes. And I took microbiology. I had to take microbiology course when I was a student, and it was a course I did not like. And I finally figured out why, because I found out I was colorblind, and you were supposed to look through a microscope and stain things and identify the organisms, and I couldn't see the color, so it was a tough course. But now I'm trying to figure out how can I learn more about them. So this is a PLFA that Dan talked about, and this is just one of those things here. We put ammonia on the field, and then I had a fall cover crop in another place on the field. But this ammonia thing, I wanted to prove that ammonia didn't hurt soil microbes. So we had a field where we split it in a cornfield, and part of it was going to be soybeans, the other part back to corn. Put ammonia on in the fall, and I took samples the first week of May for PLFA, and they had 40% less microbial biomass where I put ammonia on. And it just really shocked me, and now the tenant is using UAN solution instead of ammonia. But I want this fall cover, we had another field nearby, and the microbial biomass about the same, but the diversity index here, 1.1, is very, very poor, and the 1.6 like Dan had, where he was showing that's rated as excellent. So when we have a mix of crops out there, we can make our microbial diversity much better. And that's bacteria here. Bacteria like nitrogen because they break down ammonia to nitrate, and then we won't have to put an inhibitor to keep the bacteria from growing. But the bacteria then is at the expense of fungi. Very little fungi where we put this on, and compared, here we have less bacteria and a lot more fungi, and no mycorrhiza here, and over here we had 28 nanograms per gram. And then protozoa, we had protozoa. The protozoa they're a little bigger animals, I guess you could call them. And they go around eating all the bacteria and some of the fungi, and then because their carbon-nitrogen ratio is wider, their waste is nitrogen compounds that the plant gets in. So the protozoa are the very good beneficial to the thing. So then in the rest of that, the fungi-bacteria ratio 0.02, which is very bad, to a good level here of 0.31. And the reason we look at that, the fungi are important for making those glues and stuff and then the hyphae that kind of hold the sand, silt, and clay particles together to make the water-stable aggregates. And then you need these water-stable aggregates so water and air can flow in and out of the soil. And then the prey, we had no predator here, so you don't have near the activity in that soil. And then over here we have the predator or the protozoa, which eats those things, and that makes the soil more active. And then the gram-positive, gram-negative ratio just means it's more balanced. And the gram-positive are the bacteria that make the soil smell good when you do tillage, in the good old days when you used to smell that stuff. And then the gram-negative bacteria, or the rhizobia bacteria, is in that class. Doesn't mean anything else. The positive and negative don't mean anything else other than identification. So why the cover crops then? Recycle nutrients, like we talked about, especially nitrogen. Increase the biological activity. Competes with weeds. And we've heard a lot about that this morning. And this is the one that when I see Palmer amaranth, and I've been in Kansas and seen it really bad in soybean fields, this is one that we really got to think about. And we've been on about 50 years, probably. Atrazine came out in the 60s. Well, 2,4-D was here before that, but 50 to 60 years we've been dependent on chemicals for weed control. And before that we did lots of cultivation and tillage, but we need to now figure out how to do that weed competition with competition from other plants or other crops. And that cover crop is growing at a time when nothing else is growing to keep the weeds from growing. And I didn't show a picture, but Jody has a picture of a field planted in 2012 when it was dry, and then the cover crop was only about this tall. But there was a little space where they didn't drill, and Mother Nature had pigweed and mare's tail growing in that area. She wanted to cover there, so she put it in there, and where we planted stuff, then she was happy and didn't have that weeds growing. So the weed thing is one of those that I really think about. Increase diversity, utilizes excess moisture, and this one is the other part of the farmer part of me, decreases input costs. If you're doing a cover crop and you're doing everything the same as you're doing before, you probably can't make it pay. Because I really think that when you have a good soil life, all that stuff, you'll be able to reduce some of your input costs, your herbicide costs, maybe one spray and you don't have to do on those kind of things. So think about all those things. And of course, in the end, what we're all about and what Dan was talking about, up at Windside at that field day in June, I was able to figure out, I took the soil very carefully and I had about ½ inch of what I call new soil. So I went down to our farm and you can see, here, whoops, my thumb here, you can see that's about an inch. And I dug that up very carefully and that soil here dropped out. And these are, there's residue roots and everything, and there's little round pellets. It's kind of hard to see there, but the worm castings from the earthworms. And you think about what we're trying to do is the soil. I was talking to a farmer from Osage City, Kansas. I said, I think the soil builds at the top. And he said, well, Darwin figured that out. And he sent me the paper, 1881. Darwin said that artifacts sink because worms are bringing soil up to the top. And you think about that, a house that's 100 years old, the sidewalks sunk down in the ground, those kind of things. And when we stop doing tillage and start growing things, and you get the worm activity that Dan showed you and stuff, and now you begin, this is all new, what I call new soil, up on top. And it's just, think about 500 years from now when people are trying to feed people, the farmers are, it'd be important to have a better resource than what we had now. And it's just one of those things that I think about in trying to develop a good farming operation, so to speak. So, you guys got any questions on this? Yeah. Yeah, the fall applied ammonia, I'm saying it does set back the microbial diversity in the soil. The bacteria pick up and they come a lot more. So if you could reduce the ammonia application or stop doing ammonia and get more microbial life, then you might be able to reduce the nitrogen rate. The ammonia is a good nitrogen fertilizer. We always said those things. And what I'm finding out is if I can increase my microbial diversity, I might be able to reduce my nitrogen rate to compensate for the cost difference between those two products and still have my same kind of response. It's one of those things that from a microbial standpoint makes sense, from a farming operation probably doesn't. Yeah. Henbit and pennycress, good cover crop. I guess that was, yeah. Henbit and pennycress Is that a good cover crop? Yeah. Right. Well, I'd like to have a little diversity. If you get pennycress and henbit together, but you don't see them growing together. Yeah. In 1974, we moved to Stillwater, Oklahoma, and in the spring of '75, a lot of lawns in Stillwater were purple. And what the heck is going on? Well, henbit. Now, so I knew what henbit was, but now it's up here all over. The interesting part of the henbit pennycress thing is, in my garden last spring, I was hoeing, trying to get rid of the henbit and the pennycress, mainly henbit, like you say. And when I was hoeing that, cutting that off, it was moist where that henbit was growing, but where there was bare soil, it was hard, and I couldn't hardly get the hoe in the ground. And so they use it, just call it a moisture recycler. And if you would plant a cover crop in the fall, then I think you could reduce that population. But he said he had ideas of putting cover crop in last fall, but his combine had other ideas. And the question about planting rye, this time of year, if you can plant rye in December, it'll come up and it will be growing in the spring. So we got to rethink. When you normally think when you got cover crop, we like to see stuff really growing, but when I had wheat plants this tall, about 2 or 3 inches tall, and just a massive root system right underneath that. And the root system is what's feeding the microbes. And the tops is what feeds us. So think about that. If you have something growing, it doesn't have to be real big to be a benefit to the soil, physical, chemical, and biological system.
Ray?
Yeah.
So you had the slide about the nutrients in the above ground biomass. Do you have any idea how the above ground biomass compares to what's below ground, what's in the roots?
I do not have any idea of how many nutrients are in that below ground part. The roots there, and I haven't really tried to look at that. I did take a corn plant one time and analyzed the nutrients in the root system, and they're not quite as high as they are in the top part. And so I think the main thing the root system does is brings everything up to the top. It takes stuff down here, brings it up so it's back up on top. So when the new plants come out, and those new roots are right on top here, they're feeding all those nutrients are recycled up. And so that's what the part I worry about. Now, the reason you want good root system in the soil is because roots have more lignin in them, and that's what builds that organic matter in the soil. Because the lignin decomposes slower than the carbohydrates are in the top part. I do know that part, but that's about it.
Another question. Talking about phosphorus, decreasing phosphorus fertilization. If you're running like a soil, it's about 15 or 17 parts per million is kind of a marginal line for need or not need. But if you got a soil that's like a 10 or a 12, is the cover crops going to overcome that?
The thing that the people talk about is if you have mycorrhiza, you can get by with less phosphorus. And Dwayne Beck has been working with me or talking to me about maybe not having to put as much or not having to have such a high soil test and maintain that soil test at a lower level. I'm still old-fashioned enough. I still like that be up to 25. If I know I'm at 25 part per million on a Bray or Malick test, that I can go a year without phosphorus if the economics are bad. But if you have a test of 11 and you have a good mycorrhiza, you may not have any phosphorus deficiency. And so maybe you can maintain a lower level with that cover crop thing in there because you got more mycorrhiza. But you still need to remember that every bushel of corn takes off 0.35 pound of P2O5. Every bushel of soybeans takes off 0.67 pound of P2O5. And when you haul that off, you need to put some back, or either to maintain that soil test level where you think it should be. But you can argue all day about what that test would be, and everybody can prove you wrong or prove you right, whichever one you want. But so it's a good point. That the thing I worry about is there's people saying, well, if you have a good soil health, then you don't need to put phosphorus on. And 100 years down the road or 500 years down the road, when we're out of phosphorus, now how are those people going to get phosphorus in there? So why not have enough responsibility to maintain your phosphorus test wherever you want that to be by putting on what you're removing or part of what you're removing?
Ray, one of the sources of sulfur is the mineralization of organic matter, right?
Right.
So if we're improving our organic matter levels, are we going to correct some of our sulfur deficiencies?
It's a two-edged sword question.
I'd never do that to you, Ray.
and it's the reason we're having sulfur deficiencies, I think, is because we've cut out a lot of tillage. We're trying to build organic matter, and when we build organic matter, each percent organic matter has 1,000 pounds of nitrogen in 7 inches of soil. 1,000 pounds of nitrogen, 330 pounds of phosphorus, 130 or 150 pounds of sulfur. And understand that when we build organic matter, we have all the nutrients in there. If you got zinc that comes out of the organic matter, you destroy that organic matter. And so to build organic matter, Keith, the guys that put some animal manure on or something like that, they say they can get their soil health going faster. Well, it's because they're putting all the nutrients back in there. And I think that's so to build organic matter, we need to make sure we got all the nutrients available. And if you want to build organic matter and you take the sulfur out of the organic matter, are you going to build organic matter? No. So the the more you want to build organic matter, the more you need to put sulfur on. And it's not, that ratio of about 10 nitrogen, 1 sulfur on most of our crops would be good. On soybeans, you don't put the nitrogen on, but you could put a little sulfur on. In the canola areas or the Nebraska areas, then those crops need more sulfur. But we don't worry about that right here. Yeah.
In carbon sequestration, are cover crops going to be kind of seen as a way to enhance the ability, if carbon taxes ever do come in, that we would be able to gain some carbon tax by and still help our own crops?
I think Dan really proved that very good on his farm with his carbon that he had. Once he started doing the cover crop, the carbon come up in that soil or the organic matter, and organic matter is 58 or 60% carbon. So as you increase organic matter, you're sequestering carbon. And Dan's data shows that the cover crops increases the speed of building organic matter.
Yeah, I'm just wondering if that will be recognized as being locked in good enough, because, in carbon trading, they want the carbon locked. And so I'm just wondering if with the cover crops, they will see that the carbon being locked up.
I'll just say they better. You guys are going to be the ones that can determine that. So, very good question.
Okay, thank you, Ray, and thank you for all the good work you do for Nebraska farmers.